I would like to say that I'm very happy and honored to be invited here in this beautiful city of Rio de Janeiro. So thanks a lot for that invitation. And I'm even more happy to say that I'm not the only one involved in all the work I'm going to present to. I have many collaborators that have participated either to investigation at the mass spectrometer, but also to research proposals, sampling, strategy, and of course, I mean that to all my master, PhD, and postdoc fellows who have obviously contributed to the data acquisition. So, my research interests focus, of course, on pollutant levels in marine vertebrates, including marine mammals, but also turtles, birds, and fish. We try to understand the toxicoclimatic and transfer to offspring, and using all of that, pollutant effects on the species. You all know here, because it's obviously the topic, that the toxicity of pollutant in the marine environment is complex. So there are many pollutants involved, and they can affect the immune system, nervous system, and endocrine system. So pollutant levels in species and thereby pollutant impact depends on many factors. Of course, you have the environmental contamination, but also other factors that I call key period of life, including migration, gestation, lactation, spawning. Some of them can be also associated with fasting. So all these key periods must be taken into account before concluding on potential impact. One of the factors, one other important factor is the diet of these marine vertebrates. Diet of marine mammals and other top predators has been always a central question. What do they eat? This question, of course, is primordial because the biomass taken by these vertebrates is important. As an example, if you take the sperm whale as a single species, sperm whales consume as many squid and fish as the whole fishery industry. So yes, they eat a lot, and some people would say they eat too much. So, of course, they regulate the trophic change, and what is also my interest is that diet is a source of contamination. So variation in the diet can lead to different pollutant profiles in the animals. So there are several ways to study the diet. The more, I would say, obvious one is to look at the feeding behavior. But you will agree with me that it can be easy when you look at a sperm whale feeding on a porpoise, but this kind of protocols cannot be applied on all species. Since, for instance, at long distance grazing species or deep diving species. So conventional protocols cannot be applied for that. Stomach content analysis does provide interesting information about the last meals prior to stranding or by catch. But if it is the last meal, it means that it may reflect, for instance, the diet of a debilitated animal prior to stranding, or it will reflect, for instance, the target species of the fishing net. So it concerns DNA. The investigation of fatty acids and stable isotopes has provided recently interesting data also because it's non-invasive, so it can be applied on wide populations and not only on stranded or bycatch animals. And in the case of fatty acids and stable isotopes, they give information on what has been really assimilated and not only ingested. So for those here who are not familiar with the concept of stable isotope, let me introduce a small background. So what are stable isotopes? Isotope is the atomic configuration of a single element. If we have a look at the hydrogen atom, hydrogen can exist under proton form, deuterium form with one neutron, tritium form with two neutrons. these three configurations are isotopes of hydrogen. But tritium is unstable. You will observe some decay to helium. It's a radioisotope. Deuterium, on contrast, is stable. You will not observe any decay. It's a stable isotope. Besides hydrogen, I have interest on carbon and nitrogen, because carbon has two isotopes, two stable isotopes, I must say, which is carbon 12 and carbon 13, while nitrogen has also exist under 14 nitrogen and 15 nitrogen. The ratio between the light and the heavy isotope is an important tool in feeding ecology. Stable isotopes of carbon and nitrogen are often expressed under a delta notation. It's a deviation compared to an international standard. So I will use the delta nitrogen 15 and carbon delta carbon 13 in the rest of this talk. So why am I using them? To understand how they have been integrated in the study, you need to keep in mind two central points. The first one is that a stable isotope of a cold tumor, they are proportional to those in their diet. And then that differences in isotope ratio exist among food sources at the basis of the food chain, and that these different food sources can be reflected to the food chain. So the first tenet has been validated through many laboratory investigations using invertebrates and other kinds of models, and the second has been described in many field studies showing that depending on the producer, C3 plants, C4 plants, benthic algae, phytoplankton, the delta carbon 13 value can vary a lot. And depending on the food sources, the carbon 13 ratio will also vary in the consumers. So the kind of information we get is something like that. So you have the delta here. You have the delta nitrogen value on the y-axis, and delta nitrogen value increase with increasing trophic levels. So it's often used to give some indication about the trophic position, while delta carbon 13 show only a slight enrichment from one trophic level to the next, and so it's more an indicator of primary productivity. And this whole concept has been resumed by De Niro and Epstein in 1976 already, by this well-known sentence, "You are what you eat, here's a few per meal." So the advantages of stable isotopes is that they offer a time-integrated representation of the food rather than only snapshots of ingested food. And this representation of food, of course, will depend on the turnover of the analyzed tissue. If I take a piece of your liver, or a piece of your bone, or a piece of your skin, the delta nitrogen value and delta carbon value will not be the same, because obviously bones, blood, skin do not have the same turnover. Bones will represent a long-term life, at least it is assumed to be like that, and for instance, serum or blood will represent a short-term life. So useful to compare over when you have species having seasonal variation in the diet. And in the case of people interested in contaminants, they offer also a continuous measure of trophic condition. So the first study dealing with stable isotope in marine mammals was published in 1979 already about the marine mammals and other vertebrates from the Bering Sea. And more than 30 years later, hundreds of papers are visible not only on marine mammals but also on marine fish, seabirds, and turtles. So stable isotope has been widely used in trophic ecology, and one of the first used is foraging ecology and habitat use. Also, there's been a lot of study dealing with physiology and fractionation of stable isotope between diet and consumer species. And of course, depending on the two first axes, there is a lot that has been done on contaminant tracking. You can use them to study the trophic transfer and all biomagnification process. Also, they give information about population structure. If you combine some of the functional profile to stable isotope, you can have information about the ecological structure of the population. And also, they've been used to study niche variations. If we have a quick look on foraging ecology, foraging ecology and habitat use by the investigation on stable isotope has been one of the most common and earliest use of stable isotope. One of the first studies published by Epsom and Welsh in 1992 was dealing with the Arctic, and they showed that a general increase of carbon-13 and nitrogen-15 with trophic levels, and also that they have shown that there is 5 trophic levels in the Arctic, which afterwards was used to study contaminant transfer in the Arctic. So it's proven useful. Looking at that time, we wondered that what's happening in the North Sea. So the North Sea is a very shallow environment and with a small superficie, but despite the fact that it's a very sunny and sea, it's a very productive area with a lot of fishery, a lot of traffic going on, and some marine mammal species. So the most common is the harbor porpoise. You can also meet from time to time the harbor sea, especially along the German and Netherlands coast, but you can also meet some gray seas, and from time to time you can find some stranding of white-sided dolphins, white-beaked dolphins, and sperm whales. So the question arises about the feeding of all these species. I mean, how do they share this small environment? What do they eat? And of course, after that, how are they contaminated? So we did some stable isotope investigation. You have here on the y-axis delta nitrogen 15 value in the muscle of stranded animals, and delta carbon-13 in the same tissue. And we have noticed that basically we could observe 2 groups of marine mammals. The first one included the sperm whales and the white-sided dolphins, and the second one included harbor seals, gray seals, harbor porpoises, and white-beaked dolphins. When you compare to the fish that have been collected in the same environment, you see that first of all, that white-sided dolphins and sperm whales obviously do not feed on zooplanktivorous fish or carnivorous fish because they are on the left-hand side of the figure. But harbor porpoise, harbor seals, gray seals, and white-beaked dolphins belong to the North Sea food chain and can potentially feed on those species. So you have these 2 groups. One of them with the low carbon-13 and low nitrogen-15 value, so the sperm whale and the white-sided dolphin, are typical oceanic feeders. So despite the fact that you can see them in the North Sea and you can do some necropsy on their body after stranding, they do not feed in this area. However, other species, including the harbor porpoise, have high delta carbon-13 value and nitrogen-15 value, so they are more typical North Sea foods. We will come back later on these features to see how the profile of contaminant can be associated with this stable isotope signature. One thing that was surprising, at least for me, is that carnivorous fish and seals occupy the same trophic levels, which is quite surprising because seals are known to feed on carnivorous fish such as cod. So it means that there is some structural modification of the node. Well, we were not the only one to show that. Other studies have shown that due to intense fishery pressure, there is a modification of the North Sea chain. This gets, the North Sea gets, the length of the North Sea chain gets weaker. So still many questions on the functioning of the North Sea ecosystem. Another way to use the stable isotope is to try to see the foraging area. So we did this very exciting study on leatherback turtles. Leatherback turtles are just crazy animals. I mean, they are there for, I would say, 1,000 years, but we don't know anything about them. They just spend years in the North Atlantic, in the case of Guyana French turtles, and every 2 or 3 years, they're coming back from the French Guyana coast to spawn. Between these spawning events, we do not know anything. Where do they eat? What do they do between these 2 or 3 years? And so, with the collaboration with the Paris University, we send students there, you see here one postdoc fellow, Elodie, who is trying to collect blood on these turtles. And so she has collected blood, she succeeded, and eggs, and with this stable isotope ratio on the tissues, try to see what they were eating versus what's coming here. What is amazing is that they are they always come back on the same beach here, Walimapo beach or on the French Guyana coast. And during a spawning season, they can come back 3, 4, or 5 times and lay hundreds of eggs. So it's a very intense process during this season. And as I told you, they can spend 2 or 3 years at sea. We know that because some of them have got a transponder or tag satellites on a telemetry. So some turtles spend 2 years in the ocean, some of them spend 3 years. Why? We don't know. Another question. But we have analyzed several isotopes between all these turtles. And here, you have again delta nitrogen value expressed in Vertebrates and delta carbon 13 value. The first set of data included a stable isotope ratio in plasma. Plasma of the turtle displaying a 2-year remigration interval, and plasma of turtles displaying a 3-year remigration interval. Then you have the same kind of data, not on plasma, but on red blood cells. And red blood cells provide a long-term disintegration compared to plasma. And they differ significantly. So it means that the turtles displaying a 2-year remigration interval do not forage at the same place than turtles displaying a 3-year remigration interval. So it was quite surprising because it means that if there are 2 foraging areas, it has a lot of implication when you deal with species conservation, of course. And combined to telemetry data, we know now that some turtles after the spawn season just leave French Guyana and spread to the North Atlantic. Sometimes you can find some leatherback turtles as high as the Canada. So it's a long distance traveling. Other turtles do not go in the North Atlantic, but looks like they're just crossing here and they go eastward along the coast of Africa. So this foraging strategy lasts long enough to be reflected in their tissue. So this question, this paper, from my point of view, doesn't reply to a lot of questions, but created more questions. as often. Because, I mean, why did turtles foraging along the African coast spend 3 years instead of 2? And what is the impact of African coastal fish rates on the survival of these turtles? Interestingly, we did also metals and organochlorinated contaminants in the tissue in the eggs mainly, and the level did not differ between these two turtles, the 2 year and the 3 year turtles. But we must also admit that the levels were always very low. I mean, they feed on jellyfish, so the biomodification factor obviously is quite low. So we have just seen that there is a wide use of stable isotopes in marine vertebrates. But when it is used, I mean, the organisms we are using for this study, they can be in very different phases of life. Some of them are spawning, others are just nursing, calving. So what can affect this stable isotope ratio in marine vertebrates? That's a very central question when we want to make some relationship with contaminants. You need to know the limits of your model. And there are many limits. So that brings us to the southern axis of the use of stable isotope, which deal with physiology and fractionation. So physiology and fractionation has been also a central topic for a long time. It was one of the first studies was in 78, as we were mentioning. And therefore they showed that the carbon isotope composition of the consumer was a reflection of its diet. But also very important that the difference in this value between the diet and the tissue of the consumer was dependent on the diet and the tissue type. As I told you, you won't have the same ratio in your liver or your bone. So it means that you need to take caution when you compare interspecific variation and intraspecific variation. So this first finding led to many studies involving what we call the discrimination factor, so the enrichment of the tissue in the diet, and of course about the turnover of the tissue. So one of the models we used to study this process was the northern elephant seal. Why? Because the northern elephant seal is coming back on land to breed. So it becomes easily available for tissue sampling. You can take blood, you can take blubber, you can take some milk. So when you want to study the contaminant level of the species, the only way to get an access to them is to wait for them to come back on land, and then they just don't move. So it's very interesting. But where they are on land, that's a paradox. Where they are on land, they are either they give birth for the female, either they nurse, or they fast, or they molt. So intense physiological process. So we did a study to assess the role of gestation, lactation, and fasting on stable isotope ratio, and also, of course, pollutants afterwards. So northern elephant seal has got a very wide distribution in the Pacific. The nice thing for us, as I told you, is that they're coming back on land, and with collaboration with the Americans, we have an access to the unavailable state rivers. And mother and pup were sampled on day 4 and day 21 of lactation, beginning and end of lactation. And lactation is really a very interesting phenomenon, because you see here a mother and a pup at day 4 of lactation. And after 25 days, the mother will lose 1/3 of the body mass. But the pup will gain, we have its body mass multiplied by 4. So it's a very intense and efficient energy transfer from the mother to the pup. Energy transfer means also pollutant transfer, of course, because there is no feeding during 25 days. The mother, the female starves, not the pup. So there is a very important pollutant transfer during this period. So it's a very intense process for females. It's a bit less intense for males, but that we knew already. But I must say that some of them starve also because they spend a lot of energy trying to attract the females that do not nurse and to create harems. So they don't want to go in the wild to feed because they want to keep all the females there. So yes, it's a bit less intense, but they spend a lot of energy. You see here one of my PhD students who works in that role. You look, succeed in attracting females. So, sorry, what she did is that, in collaboration with another department, they have collected blood, milk, and blubber on these wild animals. And we believe that, of course, they are representative of the wild population. And also, we focus on less invasive sampling. And the nice thing is that all these samples are easily accessible. I mean, as soon as the mother, the female is nursing, we can approach them and collect samples. Of course, being cautious with wild animals. One of the results I wanted to show you is the delta nitrogen fitting value in serum. Why serum? Because serum is not very fast turnover. And so it reflects the diet just the day prior to stranding in that case, prior to the birth. And so what happened is that between the early lactation, open square, and late open square here, sorry, and late lactation, you can observe an increase in delta nitrogen fitting value. We expected that because, I mean, when you start, you consume your own protein, so there can be a nitrogen 15 enrichment. So that was okay. What was not okay, well, that's more another kind of question, I would say, is that some females do not show this protein enrichment because they show a decrease in their nitrogen 15 value. So again, more questions. Why do they do that? What's happening in the serum? And so, to understand what's happening, we must keep in mind that there are two big physiological states. I mean, the female is fasting, which is known to cause some nitrogen 15 enrichment, but at the same time, she's producing a very lipid-rich milk, and this milk can contain as much as 50% of lipid. And so this creates a nitrogen depletion. So the serum, it looks like the serum is not the good substrate to study the diet because the turnover is so fast during this period that you can randomly have some idea on the fasting, on milk production, but you cannot predict what will happen. Yes, I'm just saying, so the serum is react in an unpredictable way. It's because it has a very fast turnover, and it looks like under these circumstances, I mean, fasting and milk production, the half-life is not what we think. So, new question again, and no answer at the moment. So this is just a short summary of what we have here. The line of life, I would say, with the gestation and lactation. And when we sample at day 4, at the beginning of lactation, I mean, the tissue will reflect what has been occurring during the gestation. When we sample at day 21 of lactation, it will reflect what happened during the lactation period. And thus, what is happening during the gestation is mainly a transfer between the maternal serum and pup serum. So we are expecting to see a fractionation between the mother and the pup because the pup, I mean, consumes the tissue of the mother in some way, if you know what I mean. But lactation is very different because you have an intermediate, which is the milk. So we expect some fractionation between the milk and the pup. And we did observe that. there is an enrichment between, at early lactation, between the maternal serum and the pup serum, and the relationship between these two values, but not during late lactation period. And also, there is an enrichment between pup serum and milk, meaning that you have also this kind of relationship. When you get back to the pollutant load, it's also interesting to see that if you have a look at carbon-13 value here, expressed in the milk during early and late lactation, here blue, it's very similar to what has been measured in inner blubber, but not in outer blubber. So it means that the milk is produced from the inner blubber and not so much from the outer blubber. And so do the pollutants also metabolic. Other studies have shown that it's mainly the inner blubber that will be highly metabolic active and provide all the remobilization of pollutants during gestation and lactation. So yes, there's still a lot to understand. And we see that the fractionation between mother and offspring is tissue and time specific. It is important in the framework that sometimes the pup are used to get further knowledge about the foraging area of the mother. So yes, there are relationships between the value of the pup and the mother, but there are also different kinds of process and physiological pathway that should be taken into account before trying to make foraging area hypothesis. So that leads us to the trophic contaminant axis. So trophic contaminant, trophic transfer contaminant is a big topic, especially for air-breathing animals, for which the food is the main contamination pathway. So if you remember the studies we did, here you have again delta nitrogen value in muscles and delta carbon-13 value. And I'm sorry for the noise, that's possible from my laptop. So this is a value on porpoises from Iceland, Norway, Baltic Sea, that's new, Denmark, and Belgium, and Germany, of course. And the first thing you can see is that obviously the values are different from one group to the next, meaning that the foraging strategy may be different, or that the basis of the food chain here somewhere will depend on one ecosystem to the next. So we have compared contaminant value between these groups, and we looked at the potential relationship between this stable isotope value and metals, but also organic contaminants. And at that time, I was really looking forward to see, this nice relationship described in many papers between mercury, PCB, and the trophic level, and I found nothing of that. So yes, at first I was quite disappointed. What's happening with my data? And but I did find something very new for me, at least, because we find a relationship between nitrogen-15 value and cadmium levels in the tissue. Porpoises displaying low nitrogen-15 value can display very high concentration of cuteness, of cadmium in the cuteness, sorry. And it is especially true for porpoises from Iceland and Norway. If we have a look now, not only on porpoises, but on other species from the North Sea, you have again the same Y and X axis, and the two groups of marine mammals, remember the penguins and the white-sided dolphins, and the seals, and the porpoise, and the white-beaked dolphins. And we did find the same relationship. So the highest nitrogen-15 value in muscles, the lowest cadmium to cuteness. Here in this figure, you cannot see the penguin because the penguin is somewhere here, so it was not possible to put it in the figure because the concentration of cadmium in the kidney were so high. So the cadmium concentration were really, high in white-sided dolphin and sperm whales. So why are they so high? Well, it is well known that cephalopods, which is a very important point of sperm whales, they can also contribute to the diet of white-sided dolphin, can accumulate cadmium in their digestive gland, and there is trophic transfer of cadmium. So cephalopods, yes, can be a cause of this high cadmium concentration. But also, it is well known that mammals and fish from the Arctic display high cadmium levels in their tissue because Arctic is believed to have cadmium natural sediments. So there might be also a gradient from the south to the north, which would explain why the cadmium level in porpoise from Iceland and Norway was so high compared to porpoise on the southern part of the world. And as I told you, we find no relationship between mercury and delta-lateral fitting cadmium. So, question, what's happening with other pollutants? So we did collaboration with Antwerp University, and they have analyzed perfluorinated compounds. So thanks to Roland yesterday, you know already everything about that topic. The most common, the predominant compound is the PFOS, the perfluorooctanosulfonate. And they have a very unique property because they have this long hydrophobic chain, but also a functional loop, which is more hydrophilic, which is important when you deal with the beef biogas. So they have unique physical-chemical properties. And thanks to all, you know already that they can be found everywhere, they have a broad application, and of course, they can be found now in the ocean, and even in what we thought could be pristine areas such as the Arctic. They are highly persistent, and they have a bioaccumulation potential. They do not accumulate so much in the blubber, the fatty with the tissue of mammals, but more in liver and blood, because they tie to proteins. And so again, you have here my stable isotope values, so delta natural 15 and delta carbon 30. And here also we observe two groups. One first group here contains the sperm whales, again, the white-sided dolphins and the fin whales, and they have low stable isotope value and low PFOS concentration. But the second group is still the same, containing the harboseals, the white-beaked dolphin, harboporpoise, and whale. So more inshore species display far higher concentration in their liver. So here also, stable isotope was proven useful to understand the way of contamination from PFAS. With other contaminants such as PBDE, PCB, and mercury, a relationship was less obvious. We didn't find clear relationship between stable isotope and mercury, PCB, or PBDE. Third, of course, there were relationships with the biomagnification factors, so concentration in the consumer, concentration in the prey, and natural fitting value. But this absence of relationship means that there are other intra and interspecific driving factors, which include the lipid content of the tissue, of course, the age, which can be more important than the trophic position when you deal with mammals, and the sex. So many more questions to be answered about how this complex interaction can affect contamination of marine vertebrates. Another kind of relationship we have observed is that if we get back to the school female fasting and nursing at the same time, we look also at potential relationship between stable isotope and contaminant in blood during early lactation. And we didn't find any relationship except for selenium, which was quite surprising. Because selenium in maternal blood were linked to delta carbon 13 value and also linked to maternal body mass. So this surprising relationship, one more, raised question. I have more questions than answers today. Because I mean, selenium is essential element, so it needs to be metabolized, but has been previously linked to, for instance, demospongier or plaque ecosystem because they accumulate selenium. So, is there a trophic transfer also of selenium in North American fields? We need to check that because that's of course very interesting. So, to conclude, I mean, over the past few years, stable isotope ratio of carbon, natural, and other stable isotopes, because I'm focusing on carbon and natural, but we have stable isotopes of lead, hydrogen, oxygen, there are many ways to use them. They have been increasingly used to study ecological. It's interesting because, I mean, contaminant concentration in biota are affected by many complex interactions, environmental process, and also the use of stable isotope ratio can be used to understand the conjugation you have between physical, chemical, and biological factors. But, because there's always a but, there's still a lot to be done, and recommendation and future direction should include controlled laboratory studies. I mean, animal with constant diet to study the turnover link in the tissue. We are working on that. We have just finished a study about killer whales in a zoo that was on constant diet, and we have taken blood on a regular basis to see how it can be evolved, how it can change. But also, there is an urgent need to standardize the nitrogen 15 values at the basis of the food web. Because at the beginning, we were using phytoplankton or zooplankton to have the idea of the ground zero. But it has been shown, of course, that there is a lot of variation with this kind of interpretation. And so, many recent studies suggest us to skip this particulate organic matter and to use filter feeding methods, which provide a better integration of the variation, seasonal variation, for instance, of the basis of the food. And of course, that physiology, animal movement pattern, all these very intense process should be considered when you're dealing with stable isotope in the use of tracking contaminants in animal environment. So yes, once more, many more questions. So I'm taking you through the opportunity to thank all my funding agencies because we know that money in research is phenomenal. I'm thanking also once more all my collaborators here, even here in Brazil, who have put with me a very exciting study about Brazilian marine mammals. And I'm thanking you for your attention during this talk. And of course, if you wish further reading, just run and connect yourself to this website because it is the university website for all our PDFs. So with 2 clicks, you can have access to all the PDFs. Some of them have been presented here. And it's part of the open access strategy of Liège University. I'm promoting the open access side. So thanks one more for your attention.
Yeah, congratulations for your very beautiful presentation. Yeah, the point of cadmium is also always very intriguing. I wonder, I've never seen the relation with carbon, delta carbon, the team in cadmium accumulation. We always think, yeah, not only the the mammals, how they could uptake, incorporate so much carbon, usually very absorption, but also the cephalopods, how they accumulate so much carbon. Now, with this delta carbon, low delta carbon providing high carbon, would you suggest some explanation for this regarding a low diet in some essential metals that could kind of analogy of some other metal that could provide higher absorption of carbon? So I think these experiments with animals in zoo, like you are doing now, could be interesting to see if you reduce the kind of food, so option of metals or proteins or something that could change the absorption. I mean, what do you think about it?
But also, to answer to your question, yes, this relationship is quite intriguing. And going back to the cephalopods, we have also, it was not shown here, but we have also analyzed stable isotope value in cephalopods from the southern North Sea. And the stable isotope value were not those encountered in white-sided dolphin and sperm whales. So one hypothesis is that yes, there could be a cephalopod consumption, but I think it's more or less cephalopods from the northern part of the country. You know, for instance, along the Norway coast, when you have very deep environment along the Norway coast, and also a very high amount of cephalopods. So I think the decreasing carbon-13 and nitrogen-15 value reflect this more oceanic feature of the Norway and Iceland coast compared to the very shallow part of the North Sea. So well, both of them are part of the North Sea, but it's a very different environment. And going back to the in vivo experiment, yes, it could be very something interesting to try, but we need marine mammals that feed a lot on cephalopods, which is not obviously the one that are just kept in marine land or zoo. So it will be one of the next steps I would like to try. Yes.
I was very curious about the article on turtles from French Guiana, and I must confess that obviously since I'm asking, I didn't read the article yet, and I will do it. Yeah, but you said that you had some PCB data also for these individuals, these turtles. So the question is that if, I don't know if it comprised the paper also?
It's another paper, just published recently in Aquatoxics, also on OB.
Okay. Because I was curious about the use of the kind of PCB signature in these turtles, if it was possible to use that in kind of discriminant analysis on this kind of thing, in order to have a look if...
Yes, but we did a discriminant analysis between the 2 or 3 years regression interval, including PCB profiles and DDT profiles and other organochlorinated pesticides. But the difference were really it was not significant. We could see something, but it was not significant. And I think one of the reasons was because of this that's very good for the turtles, of course, but because the contamination level was so low, and it just eat jellyfish. So it could be something to try on carnivorous turtles. But for the Leatherback turtle, at the moment, it's quite lucky because the background contamination is low.
For the turtles, do you see any health issues? I know, for example, that in Australia a large percentage have cancer of turtle populations in certain areas.
For these turtles, we haven't observed any disease, but I know that there are some viruses affecting turtle population, not for the leatherback turtle, but green turtles. But for these turtles, we haven't seen any health issue. But that's an interesting question, because that's why we have also started in vivo exposure, not of sea turtle or turtle, that's not possible, but we use the in vivo model, the Florida turtle, Pachymus scripta, to try to see how, because nothing is known about threshold level of toxicity in reptiles. It's an open field for research, yes.
Thanks.